Welcome to the Creative Commons RX podcast. Before diving into this episode, I want to ensure we're all on the same page. This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.
Welcome back to The Deep Dive, where we tear through your sources, articles, research papers, curriculum notes to deliver the most critical insights right now. Today, we are undertaking a deep dive into, well, toxicology.
Right. And this isn't just the forensic science you see on TV.
No, not at all. It's so much deeper. It's the profound study of how chemical substances interact with our bodies and the absolutely wild journey it took.
The journey to transform this discipline from, ancient mysticism?
Exactly. From mysticism into a fundamental pillar of modern public health.
And that journey is what we're focusing on. Our mission today is to give you a comprehensive look at toxicology's foundations, drawing directly from professional curriculum notes. We're covering everything from its pivotal disasters, the history of poison control, right down to the fundamental principles of molecular science, the really critical toxicokinetic rules.
Okay, let's unpack this. If you boil down the entire history of this field, the core question is, how did we move from ancient figures looking for a single mythical universal antidote?
A magic cure-all.
Right, to today, where pharmaceutical manufacturers are required by law to spend years, millions of dollars, proving the safety of every new pill before it ever hits the market.
It truly begins in the dark, in what historians call the era of mysticism and superstition.
Right.
For millennia, the understanding of poisons was just. It was intertwined with magic, with religious ritual.
So poisons weren't just chemicals, they had supernatural properties.
Exactly. And the ancient toxicologists, if you can call them that, were often sorcerers or religious figures. They were relying purely on rituals, not any kind of chemical understanding.
And the ultimate expression of that faith-based approach was that quest for the universal antidote.
One cure for everything.
I'm always fascinated with things like Mithridatum and later Theriac. I mean, these were these elaborate, complex concoctions.
So complex.
Sometimes containing 60 or more exotic ingredients. We're talking dried vipers, crushed gems, you name it. And they persisted for centuries.
And they were touted as cures for virtually every poison and ailment, despite having absolutely zero scientific basis for their efficacy.
So what kept them going?
Belief, anecdote, political power, not scientific proof.
So what was the turning point? When did it start to become a science?
That happened in the Renaissance with Paracelsus. He was the pivotal figure who shifted the focus entirely to observation and, most critically, to quantitative thinking. His contribution was truly revolutionary, the dose-response relationship.
The dose makes the poison.
The dose makes the poison. That is the principle that still anchors the entire field.
A substance isn't inherently bad. Its concentration and exposure level determine its toxicity.
Exactly. And by the 18th and 19th centuries, things really started to take off. You saw the birth of toxicology as a science rooted in, actual experimental rigor.
You had pioneers like Felice Fontana, famous for his deep experimental research into viper venom, of all things.
And then, of course, the towering figure is Bonaventure Orfila, often called the father of modern toxicology.
Orfila's genius was taking that abstract science and applying it directly to the legal world, right?
That's it. He championed the use of detailed chemical analysis and autopsy findings as irrefutable evidence in court proceedings. He effectively established forensic toxicology.
And here's where it gets really interesting for me. Orfila's work meant science could finally challenge the traditional reliance on just symptoms.
Right. Before him, courts relied on circumstantial evidence. How sick did the victim look?
But chemical analysis provided objective, undeniable proof. We can see this in action with the development of the Marsh test for arsenic.
The Marsh test was critical. Arsenic was a common and historically a very difficult to detect poison.
So when the Marsh test, which could detect tiny quantities of it, was used in that high-profile 1839 Marie Lafarge murder trial, That was a huge moment.
A total watershed moment. It produced a visible, undeniable chemical reaction in court. It demonstrated the power of objective, replicable scientific proof over centuries of just guesswork.
Toxicology had officially left the age of sorcerers and entered the age of the laboratory.
But proving poisoning in court is only one piece of the puzzle. That rational science eventually had to tackle some massive regulatory shortcomings that led to real tragedy.
Which takes us to a major turning point in American history, the elixir sulfanilamide disaster.
To really get the catastrophe, you need to look at the legislative landscape before 1938, the original 1906 Pure Food and Drug Act. It was essentially toothless on consumer safety.
So what did it actually do?
It focused almost exclusively on preventing misbranding, making sure the label matched the contents, and preventing basic adulteration. It did not, and this is the key part. It did not mandate that manufacturers prove their product was safe before they sold it.
And that gap proved fatal in 1937.
Horribly fatal.
A drug company decided to market a liquid version of sulfanilamide, a known effective antibiotic for children. To dissolve the drug into a palatable liquid, the company chemist used the solvent diethylene glycol,
DEG, and they just assumed the solvent, the excipient, was safe.
which was a tragically wrong assumption.
Terribly wrong. Diethylene glycol is an industrial solvent. You find it in antifreeze and brake fluid. It causes catastrophic kidney and liver failure.
And over 105 people died,
Yeah.
many of them children, in 15 states. The widespread fatalities and the agonizing slow deaths. It just exposed the dire consequences of these inadequate safety protocols.
And the sheer lack of knowledge about the toxicity of so-called inert diluents like DEG.
So what does this all mean? The public outrage was immediate. It was immense.
It had to be. And it was the direct catalyst for landmark legislation, the Federal Food, Drug, and Cosmetic Act of 1938.
And this law, for the very first time, mandated pre-market safety testing for new drugs.
The entire regulatory focus shifted overnight. It went from just ensuring labels were accurate to legally demanding proof of safety.
But it's so tragic that it took that kind of event to make the change.
It is, and it also highlights an ongoing global problem. Tragically, diethylene glycol didn't just disappear. It has continued to cause poisoning incidents globally.
Really? Wait.
Places like Panama, India, Nigeria, often due to its illicit or mistaken use in pharmaceutical or food products. It just underscores the relentless need for international safety standards and collaboration.
Okay, let's fast forward a bit. Let's look at how the medical community adapted to the massive proliferation of new chemicals, plastics, and drugs after World War II.
Right, because suddenly there's all this new stuff in every home.
Exactly. And it became clear that unintentional poisonings, especially among children, and suicides, were major public health issues that demanded a coordinated, immediate response.
Which drove the creation of the first U.S. Poison Control Center. That was in Chicago in 1953.
Initially, these centers were purely informational lifelines, right?
That's all they were. I mean, think about it. A pediatrician in the 1950s couldn't possibly keep up with the ingredient list for every new pesticide or cleaning product or medication on the shelves.
Just impossible. Information overload.
So the centers served as rapid response resources. They'd relay crucial data to physicians in an emergency.
So standardization became vital pretty quickly.
Absolutely. The National Clearinghouse for Poison Control Centers was established under the FDA in 1957. Their job was to streamline operations, distributing standardized information.
And they were doing this on index cards initially.
On index cards, which was a huge undertaking, but it laid the essential groundwork for a national surveillance and data system.
Then came professionalization. The AAPCC, the American Association of Poison Control Centers, was formed in '58 to establish uniform standards.
And then technology completely revolutionized their capability. Things like microfiche in '72, and then early databases like Toxifile and Poisondex.
Which allowed them to access toxicity information on really obscure synthetic chemicals, what we call xenobiotics.
Instantly.
Yeah.
Which had been completely impossible before.
And today, they are absolute public health powerhouses. The Poison Control Center Enhancement and Awareness Act of 2000 secured permanent federal funding.
It led to the consolidation into dedicated regional 24-hour centers.
And established that single nationwide toll-free number we all rely on, 800-222-2222.
Now they're monitoring everything from acute ingestions to environmental exposures and providing critical data to track epidemics, like the synthetic opioid crisis.
It's an amazing evolution. To really appreciate what they do, we have to look at the underlying science.
We do. Toxicokinetics. This is the study of what the body does to a foreign substance, described by those four processes: absorption, distribution, metabolism, and excretion.
The universal ADME principle.
ADME.
So let's break down the A for absorption. When a substance is swallowed or absorbed through the skin, it needs to cross cell membranes to get into the bloodstream.
And that's why its lipid solubility is so critical. If a chemical is highly water-soluble, it often struggles to get past those fatty cell walls.
But if it's highly fat-soluble, which we measure using the partition coefficient, or COW, it just slides right through.
And we track how much of that chemical actually gets into the system using bioavailability, or F. That's the fraction of the dose that reaches the systemic circulation unchanged.
And that's often dramatically reduced by the liver, right, which acts like a kind of border patrol.
That's a perfect analogy. Before the substance enters general circulation, the liver tries to metabolize and filter it out. We call that first-pass metabolism.
Okay, moving to distribution, we encounter volume of distribution, or VED. Now, this is a theoretical parameter.
It's not a real volume in the body, which can be confusing.
Right. But it is incredibly useful for clinicians. I think of VED as asking, where does the drug prefer to hide?
That's a great way to put it. If a toxin has a low VED, it stays primarily in the bloodstream.
Which is often a good thing, because it means treatments like dialysis can filter it out more easily.
Exactly. But if it has a high VED, the xenobiotic has distributed extensively into body tissues like fat, muscle, other reservoirs outside the plasma.
And when it's hiding in the tissues, it's much harder for removal treatments to get to it.
Much harder. We also have critical anatomical barriers like the blood-brain barrier and the placental barrier, which try to restrict distribution to vital organs.
Then comes metabolism, primarily in the liver, which I like to think of as the body's water solubility factory.
That's a great analogy. The liver's whole job is to take fat-soluble substances and make them water-soluble so the kidneys can easily flush them out.
And it does this in two main phases.
Right. Phase I reactions use enzymes, like the huge family of CYP enzymes, to add functional handles to the molecule. Then phase II takes those handles and attaches large, heavy pieces of luggage. It's called conjugation.
Like attaching glucuronic acid or something similar.
Exactly, making it big, bulky, and water-soluble.
And here's the crucial twist. Metabolism is usually detoxification, but it can backfire.
It can. That process can sometimes lead to bioactivation, where the body accidentally creates a metabolite that is far more toxic than the original substance.
Which brings us to the key distinction. Toxicokinetics versus pharmacokinetics.
It's a critical difference. Toxicokinetics is the study of ADME specifically under toxicity conditions.
Getting an overdose.
An overdose. And this is so important because when you flood the system, say, you overwhelm those phase I and II enzymes, or you saturate all the binding sites, the standard processes just break down.
The body can no longer detoxify reliably.
And the chemical follows an unpredictable, often highly toxic course.
That unpredictability is why every second counts in the clinic. When a patient presents with a suspected overdose, the first and most critical priority is always stabilization.
Always. You address the life-threatening issues, the ABCs: airway, breathing, circulation, before you even try to identify the poison.
Once they're stable, the assessment focuses heavily on the level of consciousness. If the patient presents with altered mental status, or AMS, there are immediate steps the clinical team has to take.
Essential steps.
So if you ever hear about a friend or relative being treated for AMS, these are the immediate interventions doctors rely on before they even know the specific toxin. First step is a bedside rapid glucose concentration.
To rule out hypoglycemia, which can perfectly mimic a coma or intoxication.
Then they administer the standard empirical treatment, the coma cocktail.
Thiamine, oxygen, dextrose, and critically naloxone for potential opioid compromise. These four can save a life immediately without waiting for lab results.
Once those acute threats are managed, we move to decontamination.
And for acute oral overdoses, the single most helpful management strategy is often administering activated charcoal, or AC.
How does that work?
It works by physically binding to the toxin in the stomach and intestines. It drastically reduces absorption and can even interrupt cycles where the toxin is reabsorbed lower down.
But time is everything.
Everything. It's most beneficial if it's administered within the first hour of ingestion.
Now, AC is this gritty, unpleasant black slurry. So historically, manufacturers tried to improve palatability by adding sweeteners.
Like cherry flavoring, chocolate, or a sugar alcohol called sorbitol.
And here we run into another fascinating paradox. It connects right back to that sulfanilamide tragedy.
It really does. It's the lesson of unintended consequences repeating itself. Sorbitol is a laxative, often used to help the charcoal pass quickly.
But toxicologists quickly discovered a severe safety issue.
A huge one. Administering multiple doses of AC mixed with sorbitol can cause profound, life-threatening side effects.
We're talking severe dehydration, dangerously high sodium levels, hypernatremia, and even death, especially in small children.
This is a perfect modern example of the Paracelsus principle applying to excipients. Sorbitol, the supposedly inactive ingredient, became the poison due to repeated excessive dosing,
Causing severe, unreplaced stool water losses.
which is why it's now actively cautioned against for kids under 1 and those with genetic fructose intolerance. It just shows that the regulatory challenges never really end.
So as we look at the 21st century landscape, toxicology is facing massive, ongoing challenges.
Huge challenges. We have globalization, which makes tracking the production and trade of toxic chemicals incredibly difficult.
We see persistent environmental toxicity, highlighted by disasters like Bhopal and Chernobyl.
And, tragically, the devastating global drug abuse and overdose epidemic, driven primarily by highly potent synthetic opioids.
Yet there are some exciting opportunities emerging.
There are. Fields like toxicogenomics are using genetic sequencing to understand precisely how chemicals injure cells at the molecular level. It's allowing us to predict toxicity much faster.
And nanotoxicology, which is dedicated to assessing the risks posed by new materials like nanoparticles.
And all of this knowledge is accelerating the push toward highly personalized preventative drug therapy.
So if we step back and summarize this whole journey,
Yeah.
we see a discipline that started with, ancient sorcerers guessing at universal antidotes.
It was then revolutionized by Paracelsus's demand for objective measurement, solidified by Orfila's chemical analysis in the courtroom.
And then forced into critical regulatory change by the tragic lesson of elixir sulfanilamide.
The field of toxicology is fundamentally about prevention and preparation.
And that central principle remains the most essential piece of knowledge for anyone. The dose makes the poison.
Indeed. We have learned to fear the large acute dose.
But I want to leave you with this final provocative thought, building on what we know about toxicokinetics. If overdose alters standard biological processes, what about chronic low-dose exposure? What unforeseen toxic effects might be building up in our systems from the proliferation of new, complex chemicals that haven't been adequately studied under those long-term low-exposure conditions? And given that concept of volume of distribution, what does a high V tell you about where these new, untested compounds might be quietly accumulating in your own body right now, in your fat, your bone, or deep in your tissues, waiting to manifest as a long-term problem? Something to mull over until our next deep dive.
This is for general informational purposes only and does not constitute the practice of medicine, nursing, or other professional healthcare services, including the giving of medical advice. No doctor-patient or pharmacist-patient relationship is formed. Using this information and the materials linked to this podcast is at the user's risk. The content on this podcast is not intended to substitute for professional medical advice, diagnosis, or treatment. Users should not disregard or delay in obtaining medical advice from any medical condition they have, and they should seek the assistance of their healthcare professionals for any such conditions. Clinical experts created the references, content, and clinical insight. NotebookLM, a Google AI tool, created the audio content, which I extensively reviewed before release. Finally, the host states that he takes all conflicts of interest seriously. Currently, there are no conflicts to disclose. For all of his disclosures and the companies he invests in or advises, he directs users to reach out independently, where he keeps an up-to-date and active list of all disclosures.